Welcome, everyone. I can see the participants are coming in, and it's really exciting to have people here who are thinking about going to Dublin for the GSBI meeting. We're really looking forward to it, and kind of as a preliminary to this, we have this seminar today, which is going to be really fun. And so I want to give you just two things. This will be recorded, and it will be available on YouTube. And then the second thing is that there are no questions and answers as Val Behan is talking. You put them in the Q&A. There's a little Q&A box down at the bottom of your screen. And so you just put a question in there and everybody can see it. And then at the end, when Val finishes talking, we will go back and all those questions will be answered. So with that, we've got a special treat because we've got an entomologist, Dr. Monica Farfan, who is also the executive director for the Global Soil Biodiversity Initiative, who is going to moderate Val's seminar. So Monica, take it away.
Hi, good morning, good afternoon, good evening to everyone. My name is Monica Farfan, and I am the executive director of the Global Soil Biodiversity Initiative. Hey Val, why don't you turn your video on? And our special guest today is Valerie Behan-Pelletier, and she is a soil mite expert. I met Val for the first time at the summer ecology program, as many others did. Took the soil mite class and super excited to have her here today. Val, please take it away.
Thanks so much. And I'm going to share screen, hopefully. And please bear with me, people, because I'm not really adept at this. But in any case, first of all, thank you so much, Diana and Monica, for giving me the chance to talk about soil mites. Now, the title I chose, Grappling with a Hyperdiverse Taxon, is primarily because soil mites are such a diverse and vast topic. And so today what I want to do is to kind of give you an idea of who they are, where we would find them, and what are some of the traits that I think have implications for soil ecology. Now, many of you know that I've spent most of my career working on aerobatic mites. And I think this photograph might explain why. This is a photograph taken of a sample of a square meter of boreal forest soil and litter to a depth of 10 centimeters. And the boreal forest was where I studied for my master's. And there I removed the collembola from this sample, but there are 230 different species of mites represented in this sample. Incredible. When you look at it, you think it's just a pile of little seeds. And what this photo shows is the high diversity and high abundance of mites. And we have representatives of the Mesostigmata with yellow circles around them. We've got Prostigmata with blue or black circles around them, a few red circles, Astigmata, and I'm sure there's an Endiostig in there somewhere. What's striking about these soils is the lack of macrofauna. There are no myriapods, no earthworms. We do have encotraeid worms, but this is a dry sample, so they weren't extracted. The other thing that's striking is that, oh, by the way, all those little brown things and white things, they're aerobatic mites. And so, you know, that's how I ended up working my career on aerobatic mites. The other thing to notice is that the size range which is between about 150 microns to 2 millimeters, is a tenable size range. So when you look down the microscope, you've got all of those animals in, a single view. You've got the world in your view. Now we can contrast this photograph with a similar size sample, a photo taken of a similar size sample in warm temperate forests in Alabama. Again, there are about 200 or so different species of mites, but what's striking is we now have macrofauna. We've got immatures of many insect groups, Coleoptera, Diptera. We've got an encotraid. And we have loads of myriapods. Now, Wim van Putten back in 2017 called this the orchestra. And isn't that a perfect analogy? They're playing the same tunes of decomposition, nutrient cycling, and soil formation, but the players differ biogeographically and by habitat. So today I'm going to introduce you to the mite part of the orchestra. But at the beginning, why a hyperdiverse taxon? Well, mites, nematodes, fungi, insects are among those group of organisms that have an expected species diversity of over 1 million species. And of course, they're far from being described, they're far from being known. For most of them, we know nothing about them. And nematodes and mites are, are among the most poorly known of the hyperdiverse taxa. So something to keep in mind if you're thinking about what you're going to do your research on. So who are the mites? So they're a subclass of arachnids. They're chelicerate arthropods. They're related to spiders, to pseudoscorpions, to scorpions, to sulfugids. They're quite distant in the arthropod phylum from insects and collembola, which are mandibulate arthropods. There are two major superorders, and just try and keep these in mind. I know it's kind of a bit boring, but it's useful to keep this framework in mind. The Parasitiformes and the Acariformes. Now, the Parasitiformes, the big group we find in soils, are the Mesostigmata, and they're an order. Now, occasionally, you will find a larval tick in your sample. That's the order Exodida. And that's because ticks, many tick species lay their eggs in litter. And if you do find a larval tick in your sample, it's a good idea to check your own body, because you may have ended up with some ticks on you, which has happened to me. And they may be feeding on you, which has happened to me. The other big order of Parasitiformes is actually a very small group, the Opilioacarids. I say the other big order because these animals are pretty big, but they're comparatively rare, found in temperate to tropical cave litter. And so we don't get them where I am now, which is in Canada. The other major superorder in the subclass Acari are the Acariformes. And here we have two big orders equivalent to the Mesostigmata. We've got the Thrombidiformes, and in soil, we have the suborder Prostigmata. So that's the major Thrombidiform suborder found in soils. And in the Sarcoptiformes, we've got the Endiostigmata and Aurabatida. Now, Astigmata, which have evolved from within Aurabatid mites. So they're not a suborder by themselves. They are a subsection of Aurabatid mites. And this is supported by both morphological and molecular studies. So, and we can think of astigs a bit like, they're like mites, Aurabatid mites on steroids. So let me run you down through these different groups that we find in soil. So Mesostigmata, now Parasitiformes, they're the most abundant and species-rich predatory mites found in soil. They are fluid feeders. They feed on just about anything: nematodes, thrips pupa, other mites, fungus gnat larvae, housefly eggs, mealybugs. A few species are fungivorous, some are bactivorous, and few are parasites. But in general, in soil, they are predaceous. About half of the 12,000 described species are found in soil, but the true diversity is probably about at least 10 times this. They're both species rich and abundant in forest soils, abundant and not so species rich in agricultural fields, and their abundance and species richness drops off as we go to desert, semi-arid habitats. Now, we have a good idea of habitat distribution for species primarily in Europe. But beyond that, we really, we have little idea. And of course, we have no idea for all the species that haven't been described yet. Now, this is a wonderful site, and these photographs are taken from it. This has the site Chaos of Delight, and these are photographs of mesostegs feeding. So here we have an adult feeding on a nematode, here feeding on a collembolan, here completing feeding on a collembola, and here again feeding on a collembola. So again, predaceous mites. And some of them are so effective predators that they have been commercialized as biological control agents of soil pests. For example, Stratiolaelapsimitis. Some of them are known to be extremely effective controls for housefly and stable fly populations. They just love the eggs of those fly species. Now, recently, Anton Potapov and colleagues wrote a beautiful paper on feeding habits, and multifunctional classification of soil-associated consumers. And I think the key messages from that paper are really relevant. So their key message for Mesostigmata is that they, again, primarily predators, that they couple root detritus and microbial-based energy channels, and that they have high trophic positions in food webs, functioning as generalist predators and as intra-guild predators. That is, they feed on other predators. Now we're moving to a completely different superorder and a completely different order. We're moving to acariform mites and to the Thrombidiformes, and we're dealing with prostigmata. Now these are the, they are predatory arthropods in some soil types. They're fluid feeders, they're microbivores, they're herbivores, they're parasites, they're hypoparasites, and many are omnivores. 30 to 40% of the 26,000 described species worldwide are found in soil. But again, their true diversity is probably at least 10 to 15 times this, if not 20 times this. And it's difficult to define habitat distribution for any species beyond a given locality or region. Now, I tried to find some data that would suggest what their preferred habitats are. And it is clear that they like grassland soils. And probably if I had started work in habitats such as this, I would have been working on prostigmata for my career. So in bunch grass prairie from Canada, they make up 93% of the population. For mixed grass prairie and short grass prairie, in Colorado, they make up over 60% of the relative abundance. The relative abundance, is over 60% of total acri. Now, there are two figures here that circled in green that are confounding. And that is figure from Antarctic moss turf and from Dryas heath in Alaska. Now, these are published figures. And they are compromised because at the time of these studies, that is Block and Dusing-Crosley, Endiostigmata, sarcoptiform mites were considered prostigmata. So a bit of a complication. Now, as I said, they're predators, and they're both ambush and cruise predators. So here in the center, we have an ambush predator, a sita, And it's sitting there waiting for something to pass by. And in this case, it's an aerobatic mite that's gone by. And now it has that aerobatic mite, that little brown thing in its chelicera. This one here at the top left has another Prostigmata mite in its chelicera. Over here on the right-hand side, we have a cruise predator moving around, looking for something that's, let's say slow moving, or that's, slow moving like a nematode, and it will feed on it. Now, there's a large group of Prostigmata that have parasitic larvae. They're predators as adults, like this beautiful trombiculate here, but as larvae, they are parasitic. And here's one parasitizing a relative. And here we have 3 little parasites on this fly. Now, they'll parasite both invertebrates and vertebrates. And they're incredible parasites. I mean, they will even parasitize adult aerobatic mites, as you can see here. Now, this is, they're going through really, really tough integument to feed on these aerobatic mites. We don't know how extensive this type of parasitism is, but they're parasites. The key message from Potapov and colleagues is that they occupy all major feeding gills, that, as we said, predators, fungivores, microbivores, plant feeders, parasites, and some are also omnivores. And I think very key message that substantial research is needed on this poorly explored mite group. Okay, now we're going to move to the other order of cariform mites, the Sarcoptiformes. And we'll start with the enigmatic Endiostigmata. This is a small group. They live in extreme soil habitats, such as cold and hot deserts, microbial crusts, tropical grasslands, seashores, sandy soils, and deep soils. And I've chosen two representatives of the Endiostigmata. Nanorchestes antarcticus is a tiny, tiny little mite, which is actually the dominant arthropod, the most abundant arthropod at Cape Bird, the Cape Bird area on Ross Island, which, as many of you know, is also where the, let me get rid of that, is also where the McMurdo Research Station is. So this character here is among 3 species of mites found in this area, and it is the dominant one. In contrast with Nanorchestes antarcticus, we have this really bizarre looking beast found a couple of years ago by Sam Bolton. It's called the Buckeye dragon mite because it was actually collected from nutrient poor sandy soils close to the Ohio State University in Columbus, who's, I think it's whatever it was given the Buckeye, Ohio is the Buckeye State, yes. So this perfectly good mite, it's got 4 pairs of legs, extended out almost like a worm, and it's got these circles of tubercles around the body, which it can move almost in an accordion-like manner to get through these deep sandy soils. And there are other nematolipids that are even longer than this beast. Now, I should mention that Nanorchestes antarcticus is not the only mite that's iconic in the Antarctic. Probably the most iconic is this aerobatic mite, or Alascozetes antarcticus, a very big thing. It weighs about 10 times what Nanorchestes antarcticus does. And it's so common and so big in a sense, with a length of over 1 millimeter, that it warrants its own stamp from the Falklands. Now we're moving into the order Sarcoptiformes. Well, we just did with the Alascozetes antarcticus, but we'll start with Astigmata, this group that I've called, let's say, aerobatic mites on steroids, a group that has evolved within aerobatic mites. They're particulate feeders and filter feeders. They, like other arthropods, they can eat nematodes. They're thought to be effective control for southern corn rootworm, and some can eat earthworm eggs. Now, about 5% of the 6,000 described species are found in soil. They like high protein food, and in fact, cheese mites, the mite that lives on the outside and can burrow in and can feed in cheese, especially the rind, and gives a particular flavor, they are Astigmata. They're well represented in arable soils, particularly well represented in desert soils. They make up a very small component of microarthropod fauna in forests, grasslands, old fields, or tundra. Now, they exploit spatially or temporally restricted habitats, such as nests, carrion, dung, stored products, our houses. They love stored products and they love our houses. They can be root pests on things like onions. Again, the key message from Potapov and colleagues is that they can be classified as microbivores, detritivores. They're feeding on fungi, algae, and detritus, or as omnivores when opportunistically feeding on microfauna or plant tissues. Now, Robotic mites are your classic sarcoptiform mites. They are the most abundant and species-rich detritivore arthropods in most soil types. In fact, they outcompete Collembola in most soil types. They are particulate feeders, fungivorous, bactivorous, some are predators, but none is parasitic. About 95% of the 11,000 described species are found in soil, but that's probably just a fifth of their true diversity. And they're especially well represented in forest soils. And I've just circled here the numbers per square meter, 425,000 per square meter in Scots Pine Forest, 117,000 per square meter in Chestnut Forest. And their numbers per square meter go down as we get to and drop out almost completely when we get to desert soils. Their species richness also drops out when we get down to desert soils, but it can be very high in savanna type soils, as these results from Zaire show. Now, we have a lot of data on Robotic mites. They're beautiful, so people want to work on them. And thanks to Mark Moran and Stefan Choi, we have some idea about how their populations are ordered along habitats. And they took 20 sites and they separated them into modified or disturbed sites, mull sites where the pH would be neutral or alkaline, motor sites where the pH would be more acidic, and then human sites which would be at the upper end of low pH sites. And what is clear is that the density, the number of individuals per meter squared, increases along this habitat gradient. And also they very nicely broke down Robotic mites into different major groups. And so you can see, how well, let's say, Tectocephia type mites are doing in different habitats. I think this is a very useful kind of crib figure for anybody who's starting to work on Robotic mites. And repeated studies have shown, both prior to Moran and Choi and post their 2000 paper, have shown that pore volume, humus form, soil type, litter type, and complexity, especially moisture and disturbance, are the really important factors in determining the abundance and relative abundance of Robotic mites. Now, Robotic mites are the group for which we probably know the most about their feeding habits. And as Katja Schneider says, they are choosy generalists. Now, this photograph is the operational end, the feeding end of a species that I named after Dak Crosley. So this is Melanozetes crosleyi, and here we have these formidable chelicera, which are used for shredding fungi and dead leaves. We know that aerobatic mites are saprophagus, feeding on dead plant tissue. They are also xylophagous, feeding on dead woody tissue, and here live tissue, or sorry, here before they finish feeding, and here when they finish feeding, and some fecal pellets. And we know that they are microphages, feeding on fungal hyphae or spores. And they're among the groups that overcome what Patrick Lavelle calls the Sleeping Beauty paradox. That is, they feed on microbial spores. Those spores can be transported in their guts and deposited in fecal pellets as viable spores and give rise to new fungal colonies. They are scavengers on dead arthropods, and here's a cluster scavenging on a dead symphyllon. And some of them are predators on nematodes. And thanks to Annabelle Oliveira, this is a small video on Pergolumna feeding on nematodes. Yes. So it's searching. It's found one. And now we have, the nematode does not want to be eaten, of course. And, but we've got those chelicera working, getting that nematode into the gut of this, this mite. And you can see it's pretty much like slurping up your spaghetti as if your spaghetti is live. Okay, who will win? Yes, the mite has got it. And now it's going to use its palps to search for another prey. And, oh yeah, okay, it's got one. And the tussle between these predator and prey will continue. Now, probably the most formidable thing about aerobatic mites, from my point of view, is that they produce solid feces. They are particulate feeders. And so when they have fed, you have these fecal pellets left behind. And we can see them here on this, this leaf that has been defenestrated. You can see them here in wood. As this nymphal mite feeds forward, it is depositing these fecal pellets behind it. And here is an adult in the process of depositing a fecal pellet. And these fecal pellets are shredded fungal tissue, plant tissue, whatever. And so you have increased the surface area for access by both bacteria and fungi. Now, these fecal pellets can be so numerous that they will make up a layer in soil. And there are fossil records of fecal pellet layers from back about 25,000 years ago. And extraordinarily, there are fecal pellets in these slices of plant tissue from 300 million years ago. So we don't know what the aerobatic mite was that was feeding on this plant tissue, but we know it was there because the fecal pellets are still there in fossil form. So the key message from Potapov and colleagues is that they belong to 4 and more trophic gills, that they are, excuse me, and this has been indicated by stable isotope analysis, that they include lichen feeders, primary decomposers, secondary decomposers, and predator scavengers. And very importantly, that classification into trophic gills above the species or genus level is not reliable in most cases. And that key message is probably applicable to all mites. Now, what I'd like to do, sorry for this, it's a table. What I'd like to do is to look at some other traits that I think are very relevant to studies on soil ecology. And these are life cycle, reproductive mode, dispersal, and defense. And I'm going to combine life cycle and reproductive mode. Now, Mesostigmata can go from egg to adult in 2 to 21 days. Now, not all of them, of course, and many of them, if the weather is terrible, they will take breaks, but they have a short developmental time. And when they reach adulthood, they can have a burst of reproduction and then die. They are semiparous. They exhibit R-style life history traits. Something similar for Prostigmata, they can go from egg to adult in 2 to 140 days. Again, short adult life with some exceptions, and some of them, many of them, exhibit R-style life history traits. Astigmata, egg to adult in 4 to 28 days, short adult life. Again, semiparous, they reach adulthood, deposit their eggs, big reproductive burst, and then die. They exhibit R-style life history traits. In contrast, Robotic Mites, by the way, from which Astigmata are a subgroup, Robotic Mites take a minimum of 23 days and up to 1,800 days to go from egg to adult. And the adult in temperate areas lives from 1 to 2 years. I mean, an extraordinary length of time for something that's pretty tiny. So we can look at this in these two figures, for which I can thank Roy Norton. So for mesostigs, astigs, and prostigs, we've got a high rate of secondary production, we've got high fertility, we've got rapid development because of this egg to adult in 1/2 to 20 weeks. They have a short adult life usually, but because of these case-style life history traits, they can respond rapidly to resource inputs. So if there's a new dung patch, if there's a new area of decaying vegetation, mesostigs and prostigs and astigs can, if they get there, can do well. In contrast, Robotic Mites have a low rate of secondary production, low fertility. Once they get to be slow development, when they're adults, they produce an egg, okay, keep on feeding, produce another egg, keep on feeding. Very few species have more than 4 eggs. They've long adult life. They've egg to adult in 4 to 260 weeks. And as a result of this, you have stable populations of Robotic Mites in your soils, but they do not respond rapidly to resource input, I can tell you. Now let's look at dispersal, another trait. So mites do not have wings. And so to get from one source of food to another source of food, obviously they can walk, but Mesostigmata are pretty speedy, but certainly astigs and Robotic Mites are very, very slow walkers. So most of dispersal is going to be by air, blown around by air, on water, getting onto birds or mammals, or getting onto moss surfaces or leaves for that matter. And where Robotic Mites are concerned, there are 146 species of birds that have been shown to carry Robotic Mites on them. But for the other 3 groups, the main thing is that they have a phoretic stage. And usually this is the deutonymph. So in Mesostigmata, the deutonymph, that is, so that's a nymphal stage, they have a phoretic relationship with the many arthropods. And here's some examples. So here we have phoretic deutonymphs getting their ride to a next resource on a beetle. And these are uropodid deutonymphs. And here we have a cluster of deutonymphs getting a ride to the next resource on a lantern beetle, and here a cluster on a bee. Now, sometimes there can be so many phoretic deuterims getting carried along. So this is like an Uber or a bus that the vehicle can't lift off the ground. Astigmata have a highly unusual deuterynymph, which is called the hypopus. In general, it's a non-feeding stage. It's inert, and it's highly resistant to any type of desiccation. And here we have some of these being phoretic, being carried along on this beetle. And now, in contrast, our batid mites primarily disperse as adults. We have no, I can't think of any situation where they disperse as deuterynymphs, except maybe here, where we have Archaegozetes deuterynymphs being carried on this opilionid. And this is our classic orobatid phoretic adult. It's called Mesoplophora, and it's basically like a clothespin grabbing onto the tarsus or a hair of a beetle. But we have big difference in dispersal effectiveness, with prostigs, mesostigs, and astigs being much more effective because they have a phoretic stage, or one of their stages is able to be a phoretic. Now, the last trait I'm going to talk about is defense. Now, if you're a predator, you really don't need much defense. It's everything you're preying on that needs defense. So Mesostigmata do have shields. They have dorsal shields, which I'm sure protect them against predators. They also have what are called pedofossa, and these are depressions in their bodies, in their sides, where they can tuck in their legs. Prostigs are most vulnerable as immatures, and they use silk to protect their eggs and immobile immatures. Astigmata have gland secretions, but it's unclear whether these are primarily, whether these are for defense or whether they're primarily pheromones. And our batid mites, well, they really need defense because we've got an adult life of 1 to 2 years. Now, you can hide if you're an immature. You can be protected in micro niches in soil. But if you're an adult and you're trying to find another adult and you're moving around feeding, you need to have defenses. And they are both sclerotization and gland secretions. So when we look at these absolutely glorious pictures of our batid mites, these are defense mechanisms. The long seta, the shield-like seta, the raised seta, like the raised seta, and these layers of protective shields are protections against mesostigmatic mites. So they are, they absolutely need these because they're slow moving and they're around for a long time as adults. Now, this is a defense mechanism, and this small video from Roy Norton will show you how it works. So here's the mite with its seta in a relaxed situation. And now it's going to be, oh, it's approached by Calemula, but that wasn't too bad. But you can see the seta raise up. I suppose it's telling everything that touches it, I'm much bigger than you think I am. I'm a bigger mouthful. Now that was a predator. The mite stopped and raised those seta. And here we see it from the back end. Those seta raised up, really impressive. Okay. As said, our batid mites also have gland secretions, and these are a complex chemical defense system. And they're coming from or produced by the opisthonodal or the opistosomal glands that are at the back end, pair of glands at the back end of the body of most groups of our batid mites, other than pretty basal groups. And there is an extraordinary array of chemicals that have been produced from these glands. Here we just give you, monoterpenes or aromatics, aliphatics, aldehyde, ketones, fatty acids. In one group, and this is a representative, we have these, we have aromatics, we have hydrocarbons, such an array of chemicals that are produced. And this work has been going on since about 1995, and is really quite an extraordinary array of chemicals. And aerobatic mites also produce alkaloids in their as part of their defense. And over 80 alkaloids are known from aerobatic mites found in Panama and Costa Rica. And so the question is, are these used by their predators are they used by their predators in defense? So we know that in these areas, the strawberry poison frog, this character here, that is the basis of the poison that's used in those poison darts, feeds on both aerobatic mites and ants. And 42 of the alkaloids that have been found in aerobatic mites have been found in this strawberry poison frog. So it's very likely that aerobatic mites are the most important dietary source of alkaloids for strawberry poison frog, and probably for the whole group of poison frogs, and possibly for other predators of aerobatic mites, such as the salamander. And just think, recently hydrocyanide has been found as released from aerobatic mites. I mean, hydrocyanide, that's a pretty toxic defense mechanism. Now, of course, defenses don't always work, and there are some beetle taxa that are effective predators on adult aerobatic mites. And here we have this particular species of the schidmanid beetles, which really enjoys sclerobated and opiate aerobatic mites. And in this photo from that study, we have the adult beetle feeding on an adult opiate mite, and you can see it's staying well away from the back end of the mite, where possibly some defense secretions would be coming. So let's take this cluster of traits and bring them into our soil situation. And I really like this study on cadavers as decomposition, as decomposition sites. So this is a study from Rai et al. In 2021. It was done in England. It was a study done over 3 years. And these authors used pigs' cadavers basically as litter bags. So they followed these pigs from the fresh decomposition stage to bloated, to active, to advanced, and then the final stage of them being dry. Excuse me, I'm just going to bring that over there. Now, and of course, each stage had a control, and the pigs were all treated similarly. Now, what's really struck me about this figure from this study was that the predominance of Mesostigmata in the different stages, Mesostigmata in black, even though the control sites had very few Mesostigmata. And the authors broke their study down into families of mites, and the families of Mesostigmata are dominated by species that are phoretic as deutonymphs. And here we have phoretic deutonymphs catching a ride on the bus that is this fly. And so that capacity to disperse as deutonymphs to a new resource is fundamental to why Mesostigmata do so well on those cadavers. Now, this is a figure that most of you are acquainted with. It's basically the shortgrass prairie food web from Hunt et al. 1987. And I've just added pictures to it to jazz it up a bit. But this is fundamentally this shortgrass prairie food web. So down here at the bottom, we've got aerobatic mites, or at least, sorry, closer to the dead or organic inputs. We've got aerobatic mites feeding on fungi and bacteria. We've got Astigmata feeding also on fungi and also dead plants, and feeding wherever there's a cluster of resources, we're going to have Astigmata. Further along this food web, we've got predatory prostigmata, which are going to be feeding on nematodes. They're going to be feeding on mites, going to be feeding on colembala, and then as top predators in this system, I'm sure there are other top predators, we have the mesostigmata, and they are feeding on prostigs, they're feeding on insect eggs, they're feeding on nematodes, colembala, etc. And we know from multiple studies that a food web such as this contributes to the ecosystem services of soil formation, the aerobatic mites, nutrient cycling, just about everybody, and disease regulation. The predatory mites are controlling plant-feeding nematodes, controlling and probably other diseases. Excuse me. Now, what happens when we really disturb that soil? So let's say we go in there with a rototiller. What happens? Well, we're going to knock out most of the predators. They're just not going to, they're going to be killed off. And certainly the aerobatic mites will be killed off because they don't like that level of disturbance. Of course, some of the colembala and nematodes will be hit as well. The astigs will probably still be around. But we know that our ecosystem services are affected. We know that we're going to have reduced nutrient cycling. We're going to have no disease regulation, or very little disease regulation, because our predators have dropped out. And we're going to have no soil formation because our aerobatids and many of our colembala are dropped out. Our system will have low resilience. And the question for soil mites is, how long does it take for them? How long does it take for the system to recover? How long does it take for them to move back into the system if it remains undisturbed? And there are a number of studies that have looked at this. In particular, let's say Adel et al. From 2006 in Georgia in no-till sites. But combining all the studies, it takes mesostigmata and astigmata about 2 months to reach back to the abundance that we had prior to disturbance. It takes prostigmata anywhere from 2 to 13 months to get back to abundances prior to disturbance. Aerobatic mites, because of their traits, life history traits, it takes a minimum of 12 months and probably up to 5 years, and in one Russian study, up to 40 years to get back to pre-disturbance level abundance and species richness levels. Quite extraordinary. So we need to keep in mind these differences in traits between the soil mite groups as we're working in soil ecology. Now, where are studies going in the future? There are two things that really came to mind. One is soil bomb, and the other is the explosion in molecular studies. So as many of you know, soil bomb is a global monitoring of soil animal communities using common methodology. And it's quite extraordinary, the scope of soil bomb. But fundamentally, it's looking at all the components. It's hoping to look at all the components that contribute to soil health. And most importantly, it is looking at these from a global perspective. with the idea of a common methodology, whether you are in Canada, Africa, Asia, wherever. And there is a growing body of researchers contributing to this soil bomb idea, which is pretty fantastic. The other thing that I see is molecular studies, because at the outset, I had these question marks on this figure. And these question marks refer to the fact that we actually don't know which group of arachnids, or at least let's say there's no agreement on which group of arachnids acari are most closely related to. And also there is disagreement on whether Parasitiformes and Acariformes are sister taxa. That is, are mites, acari, monophyletic or diphyletic? Are Parasitiformes more closely related to some other group of arachnids? Acariformes more closely related to some other group of arachnids than each are to each other. And there have, since 2004, there have been a range of molecular studies, many of them partly addressing this question. And this was one from 2015, looking at nuclear ribosomal genes, 228 taxa, and it gave strong molecular support for a non-monophyletic acari, with Acariformes more closely related to sulfugid arachnids and Parasitiformes to, we don't know. Okay. Now, recently, colleagues at Agriculture Canada, the Canadian National Collection, Marla, Monica, Wayne, and Fred, and other colleagues around the world have been using ultra-conserved elements in the genome to attack this question of, well, both within taxon relationships and also the question of monophyly of acari. And ultra-conserved elements are kind of the latest in the molecular toolkit. And they can be used to study both deep and shallow phylogenetic divergences. Now, the big difference with this study is that they're dealing with 500 taxa, just an enormous number, and loads of Acariform families represented, loads of Parasitiform families represented, and 11 different orders of non-mite Chelicera. Now, the first iteration of the cladogram was presented 4 weeks ago at the International Congress of Ecology, and I can't read it, you can't read it. It's a work in progress, but it is addressing this fundamental question about what are mites. As Fred said at that talk, yes, there is 100% support for Parasitiformes being monophyletic, 100% support for Acariformes being monophyletic, but is acari monophyletic? Are these their closest relatives? It's still maybe, maybe not. So, and of course, every time one of these is studied, it's adding to the body of data on mites in GenBank. Now, if you're remotely interested in working on soil mites, I really suggest that you start with Dave Walter and Heather Proctor's book on mites, ecology, evolution, and behavior. It came out in 2013, and it's totally it's wonderful. If you're interested in taxonomy, of course, there's the Bible, the Manual of Ecology, which came out in 2009, and many of us have chapters in it. And Zoe Lindo and myself will be coming out with a book on Robotic Mites only at the end of this month. And with that, I'd like to thank, these are the mite characters who are there whenever I have a question or wherever I'm insecure or whatever about mites, and my sister, who's my fiercest critic. And I am so looking forward to seeing you all at the GSBC in Dublin. First of all, because it's going to be an incredible opportunity to be introduced to see what's going on vis-a-vis soil bomb. Number two, because you will meet and mingle with so many people that are at the forefront of soil ecology and soil mite, excuse me, research. And number 3, because Dublin is a brilliant city. It's where I studied, it's where I grew up close to Dublin, and where I went to school, and it's just so much fun. So I'm expecting to see you all there. And with that, thank you so much.
Oh my goodness, thank you so much. That is wonderful, Val. We have so many questions for you coming in. If you can stop sharing your screen.
Okay, let me see where sharing screen.
If not, I can stop it for you.
Sorry, new share. I think it's this one, yeah? Yeah? No. Pause share, excuse me. I've paused share. Oops, not. Okay, is that okay?
Yeah, we still see it.
Oh, Lord. Sorry.
That's okay. I think I can.
Can you maneuver around that? Because stop share. Okay, there we go.
There we go.
Oh,
Thank you so much.
thank you.
Oh my goodness, Val. So we have so many questions for you.
Frightening.
Are you ready?
Yes.
Okay, great. How I think this is an important question that you know, we think of even when we're estimating, you know, insects and arthropods in general. But someone asks, how can we reasonably estimate that 80% of orbatid mites are undescribed?
It's, excuse me, sorry. Do I answer live? Yes, okay.
You just answer.
Okay.
Answer here.
This
Yeah,
is,
we're good.
yeah, so a very useful thing to consider. And what we're basing this on is our own research. And so, and also what molecular research is showing about the number of or closely how one species that we know on the basis of morphology ends up being 5 or 6 species from a molecular standpoint. And this is, I think, you know, this is really, I would say that the numbers we give at present, 1 million species of mites, for example, as an estimate, is way under. And to give you one example, I did my PhD work in the Arctic and on mites, and I came up with something like 480 species of mites as being the biodiversity in the Canadian Arctic. In 2012, I joined Monica Young in a study on a molecular study using cytochrome oxidase 1 in a single site in the Arctic. And she came up with over 1,000 species of mites based on molecular work. So this is on what I would base the figures I've given.
Got it. Okay, yeah. So molecular, especially with something so small and so cryptic, right? Molecular is what helps us to determine that. All right. So our next question is, what resources would you suggest for getting into soil mites? I think you gave some really great ones. For someone who is, you know, going out there and sampling soil and then has all of these mites, what's your recommendation? I was one of those people in graduate school, so it's, you know, it's a daunting task, right?
Yes, it is, and it depends on where you live. Now, if you live in England, Felicity Crotty gives, I think, an online course, or maybe it's strictly offline. But Felicity Crotty is who I would go to for identifying mites. If you are in, I would certainly do a Google search for mite courses globally. The acarology summer program is no longer working, but however, Arkansas University has a program on identification of soil mites. And I know that there has to be a course at Montpellier in France, and I don't know the title of it, but the French have had a long-running course since I think probably the 60s. So there are resources in terms of keys to species. Yes, there are very few things online, but I would start with a manual of acarology, if it's in your library, to get you to family. And then once you have a family, you can plug that into Google and search for PDFs that relate to that family. And you'll probably find a PDF that has a key to the genera or species in that family. And for example, let's say, yeah, that's the approach I would take if I was starting from the outset.
Okay, great. So we have a few hellos, Tom Bolger, and also Heather Proctor say hello. And Tom says, look forward to seeing you in Dublin. Okay, so we've had a couple questions about Robotic Mites as possible indicators for soil quality, good soil. What are your thoughts on that and its feasibility and use for that purpose?
Okay, in 1999, I think I wrote an article on Robotic Mites as bioindicators in agricultural systems, I think it was. And at that time, I was very ambivalent because they will tell you, let's say, if you have large numbers and high abundance, high species richness, you're probably in forest soils, but you already know that because you're sampling in forest soils. I think what our Robotic Mites are probably more useful for is for detecting high levels of certain chemicals. So especially Russian authors have shown that, Russian and German authors, I should say, have shown that they're good indicators of things like cadmium, of zinc, and a few other chemicals like that when they're in high abundance, and they respond negatively to those. But as general bioindicators, it's first of all, it is, what do you want to know? Do you want to know, is this being polluted, or do you want to know if this has is just a forest, which you can see all by yourself? Mm-hmm. A couple of our Robotic Mites are, in fact, a few of them are now used as, they are certified as bioindicators. And one of these is Oppia nitens, which is used in soils that have been affected by toxic ingredients. So, for example, oil spills. And Oppia nitens is an ISO certified mite, along with Falsomia candida, and I think one of the earthworms that is used in these studies. And there they have very precise protocols for how this mite, how you rear this mite, first of all, in these soils, and how it responds to various high levels of petrochemicals, say, in soils. So I would probably start, if I was doing this now, writing a paper now, I'd probably start with Oppia nitens and maybe Platinopterus punctatus, which has been used as an indicator of high chemical contents in soil, and then look at the references in papers of those studies and work backwards, as it were, to see what for the particular situation that you're asking about bioindicators. Okay.
So would you consider Akari keystone taxa?
Well, I do, not only because I work on them, but because they're just about everywhere. So you go into habitats. I mean, if you're up in the Arctic or subarctic or boreal, you are not going to be, you won't have earthworms. You'll have enchytraeids, but you'll always have acari. If you're in the Antarctic, in fact, I think the only place that you won't find acari are in the dry valleys in the Antarctic. So astigs have been found as far north as the furthest north land in the Arctic. So as keystone species, to tell you that, yes, some kind of, let's say, decomposition is going on. In the case of the acari in the Arctic, is because people had left some food outside their tent or something like that. But it still was a resource that acari that had astigs that had gone up with them could take advantage of. But yes, I do consider them keystone species. And I think keystone species, we always want to think of the habitat that we're working in, or the, so for example, if you're dealing with deep soils, yes, you really have endiostigs and nematodes are probably your keystone species. If you're dealing with desert soils, again, endiostigs and let's say crust soils in the Joshua Tree forest, you're going to be dealing with certain species of prostates. You're just not going to get some of the big keystone taxa.
Okay. So there's a question here that I'm going to modify a little bit. Do you think it is feasible to be using soil mites in studies of not soil quality? So this is kind of something we've touched upon, but
yes.
Is it okay? and how so?
I would say that we're well, certainly the ISO certified. The ISO certified. Just a second. There's somebody trying to. Okay. So the yes. Sorry, I'm sorry. That was a deflection. I have somebody at the door, and for soil quality, yes, of course, because we are using them already as detectors of soil quality in soils such as that have been contaminated, where you and I'm trying to think, and we're using them as it were moderators of soil quality in general when we say that a soil is rich in mites. So that's a bit general of a question. We can do it specifically vis-a-vis the species that we know are that are defined for detecting things such as heavy metals. Okay. Sorry for that. I had a disturbance in the background.
Yeah, no worries. We've got still a bunch of questions too. So Stephanie asks, a common amendment used when rebuilding soils after mining is a mix of peat and most recently biochar. What are your thoughts about amendments, these additions, and how they could affect ore-batted mites in reclaiming areas that are lower quality, polluted, mined?
any amendment that's going to provide organic material for soil decomposers is going to be welcome. And so a study that that we did, actually, I thought I was going to say Diana was involved, but no, one of her students was, Mark St. John, in 2002. We studied reclamation of mining sites at Sudbury, Ontario. And certainly the soil that where amendments had been put on certainly recovered overall faster than, now I can't remember what the amendments were, but they recovered better than those that were left without amendments. So I think which amendment works best is, of course, a subject of research, and it depends on the situation, because mining soil, let's say mine tailings, are not all the same because they have different concentrations of salts, etc. What blew me away about that site, and this is an apropos, is that in the first year of study, the only taxa that we were getting from the mine tailing sites were nematodes and Endiostigmata. It was like being in the perfect predator-prey situation. And then, of course, over time, I think it was about 30 to 40 years later that we were really getting good numbers and good diversity of our robotic mites. Anyway, sorry. Yeah.
Okay. So another question that we have, this has to do with what you told us about the fecal pellets of Orobatida.
Yeah.
And someone mentioned that this seems counterintuitive, that the fecal pellets, that they would be almost compacted as opposed to giving more surface area. And do you have some clarifying
comments,
thoughts on that? Yeah.
Now this is you know, because nobody goes down, and or at least nobody in the last 50 years has gone and teased apart a fecal pellet to measure out the surface area. But this was actually done in the 30s by an American called Jacob. And what he, and I'd have to search to find the publication, but what he estimated was that on average that there was an increase in surface area of 20 to 30%, just because you had taken something that was defined, that flattened leaf, you know, parenchyma tissue, whatever, and you had torn it apart. You had made this woolly ball of material. And of course, we're dealing with bacteria and fungi, which can get into just about anything. So we have broken up this habitat of that piece of leaf or that woody tissue and provide more access, more surfaces. Obviously, the volume hasn't changed, but there are now more surfaces for bacteria and fungi to access. But nobody has done this study since, which is a pity.
Yeah. So we've got some questions about orobatids and their phoretic capabilities. So someone says that they have seen orobatids be phoretic on lycosid egg sacs. First of all, is that phoracy? And second of all, what are your thoughts on what that might be?
Well, I suppose the lycosid egg sac isn't going to be flying off anywhere or moving off anywhere. So it's I would say it's not exactly phoracy, but so lycosid egg sac is presumably a stable thing in a particular situation. And some orobatic mites are known to feed on the egg sacs of hemlock woolly adelgids. And what they're doing is they're feeding on those waxy excretions on the surface of the eggs. And it's possible that that is what's happening with the lycosids. And if it is, and that's a good subject of research to find out whether that is the case. But I would not say it's foresty.
Okay. How would, let's see, let me amalgamate a few here. Do you know how orobatids respond to, in an agricultural system, no tillage?
No tillage, of course, they love no tillage. I mean, in comparison with tillage, because a key thing to remember is that, and just beautiful studies by Mark Moran over a period of time show that orobatic mites do not like disturbance. And that disturbance can be from any source. For example, earthworms moving in soil disturb orobatic mites. So if you have lots of earthworms, you're going to have much less orobatic mites. So your populations are going to, of orobatic mites, are proportionally to other mite groups, are going to build up in no-tillage systems. And probably the best paper that shows this quickly is that of Sina Adel and Dave Coleman, and I can't remember the third author, from 2006, and I think it's in Applied Soil Ecology. And it shows that it's looking at no-tillage systems over 25 years, and the proportion of orobatic mites increases along the 0 to 25-year gradient.
Okay. We've got a few questions about things in soil and orobatids' reactions to them, I will say. You touched upon cadaver research, and someone is asking, well, what about mites as important indicators for forensic science? Next to, I mean, a lot of people use fly larva flies as also an indicator of how long a cadaver has been out and decaying. What do you think about using mites as a similar indicator?
Well, they're already used. So
Okay.
Astigmatic mites are probably used almost more than flies, because they actually they stay around longer. So they're kind of they stay around longer, and as long as there's a food resource, we're going to have astigmatic mites. It's a bit like in the high Arctic, where you have them if somebody's dropped a piece of food. And so cadaver research, both humans and any other type of cadaver, if you plug into Google cadavers and astigmata, you'll get a wide range of publications. And I'm trying to think of the archaeologist who did a lot of this work back before he retired. But in any case, astigs are what you're looking at vis-a-vis the stage of decomposition for cadavers.
Okay. Moving along, someone wants to know is there any association with fungal fruiting bodies and orobatids?
Yeah, there's again, there are a number of studies that have, yes, a wide range of studies that have looked at different fruiting bodies. And these studies go back to the 1960s. And there was a series of studies back then looking at the fungi on on white birch. So the pore type fungus, bract fungi, excuse me. Had a mental fog there. So bract fungi. So a series of studies, and the key author there was a guy called Matthewman. So as I've said it, Matthewman, And if you look at Matthewman and bract fungi, you will come up with the studies that he did on those. Subsequent to that, there have been a number of studies in this century, studies especially out of Ireland. I think one of Tom's students looked at aerobatic mites associated with the general fungal material. And so, you know, that's another one where there are loads of references. They're just not at the top of my head. But if you go in fungal fruiting bodies and aerobatic mites to a search engine, you'll come, and PDF, of course, you'll come up with a range of PDFs. And the same thing applies to mesostigmatic mites, because there are some mesostigmatic mites that are actually modified for living in those pores. And so again, you can go into a search engine and come up with interesting studies. Okay.
How much do you know? Well, so orbatids, there are many different species. You know, we're talking about how diverse they are. In terms of their movement in soil over, I don't know, whatever measure you want to use, over their lifetime, how far do orbatids travel on their own?
Well, okay, so first of all, let's take aerobatids. And so it's been a wonderful study by Fecal Pellets and a student worked out that aerobatids over a 30-year period, I think, would walk about 20 meters. Okay, so you definitely wouldn't want to be waiting for them to join you. And but studies looking at aerobatic mites within the soil profile, especially by have shown that some species will actually go down vertically in response to lower soil moisture in the upper levels. Some species you will have aerobatic mite, you will have immatures primarily at one level and adults primarily at a higher level in the soil. And for that kind of research, I would look at papers by Malcolm Luxton and vertical distribution of aerobatic mites. Now, that's aerobatic mites, because of course other mites go down deep in soil. Aerobatic mites don't go very deep in soil. The ones that go really deep are the Endiostigmata, and they will go really almost, well, they'll go right down to where plant roots end, because of course plant roots are at their bottom ends are going to be exuding nutrients just as they are at a higher level in soil, and there are Endiostigmata species that will feed that deep. Also, many mesostigmata will move through the soil profile depending on the humidity gradient. And some of them are actually quite flexible. Their dorsal plates allow a certain amount of, sorry, I can see it here, a certain amount of movement through soil. You know, they're flexible. I couldn't tell you exactly how deep. I think there have been mites, they would have been called prostigmata, but they were probably Endiostigmata that have been found down around 15, 20 meters in soil where roots end up, basically.
Okay.
But I should say that all mites, prostigs, mesostigs, aerobatids, are going to move in the soil profile up and down. And it's very much species specific what is going on. And so it reiterates what Potapov and colleagues were saying. We need to be doing our identifications at a minimum at the genus level and preferably at the species level. Okay.
Easier said than done sometimes.
Oh, I believe me.
So we're coming close to the end of our time. So I wanted to ask a few sort of tie-up questions. One is from Stephanie, for passionate scientists who love mites, and are just getting started in their careers, how can we contribute and help grow the collective knowledge of soil mites?
Oh, what a marvelous question. Oh, what a sweetheart. I mean, how fantastic. Well, I would say, depending on the group that you're interested in, because it's very unlikely that you're going to end up working on all soil mites. It's a pretty daunting task. But if you are making sure that your research is shared with other mite colleagues, I think is very important, because very often you're working on a soil ecology problem and you're going to soil ecology conferences, but if you come across a paper in, let's say, taxonomy or whatever, or physiology, that's relevant to your study, reach out to those colleagues and start to form your own community of shared interest. I know, I'm in the Robotic community, and it's a super community. We have shared interests and shared whatever blogs or whatever. But if you're, I think making sure that you share your work, and of course make contact with, if you're, let's say, if you're in Canada, make contact with the people at Agriculture Canada, the colleagues I described. If you're in the States, well, knock on Dak Crosley's door, and may not want that, but ask help, reach out, never be afraid. It's like all soil ecology, it is a community. And of course, you're now able to contribute on an enormous scale through something like Soil Barn. And I know, working with Diana, through her, I've been involved in a couple of global initiatives. And it's just so enriching, because you are treated to colleagues that you would never meet otherwise. So get involved. Okay.
And come to the Acrological Society of America meetings.
Exactly.
Where we meet with the Entomological Society of America. So if you go to that meeting, we're there as well. Hey, one more question. I'm sorry, everybody, we're not going to be able to get to all of your questions. So apologies. We have had an overwhelming response to your talk, Val. So last question from Andres. Parallel to my PhD in Europe, I'm trying to lead a group of orobatid specialists in Colombia. Do you have any suggestions to motivate new students to get and stay in the field?
I would say just be yourself. Enthusiasm drags people in. Just looking down the microscope. I mean, how did any of us get hooked on soil mites? We looked down the microscope one day and realized that we had the entire world of dinosaurs moving about, in our Petri dish. And then you've got these Mesostigmata coming by, and they are more effective than any cheetah in the above ground world. And then you can, that's looking at it from a soil perspective. But if you're working on a group of mites, you've got such a world. If you're working on Mesostigmata, well, they're associated with all kinds of vertebrates. You've got the ones that get moved around in the nares of birds. Think about the work on hummingbird mites and their mite associates. You have this, it's, what can I say? It's a fantastic world. So your enthusiasm and a focus on a range of questions is, of course, what's going to keep students hooked.
Great. Val, thank you so much today for sharing your work with us and for your wonderful thoughts on all of our questions. I'm sorry if I wasn't able to get to your particular question if you were in the audience. Any last words for our audience, Valerie?
If you go to your library or go online and check out Dave Walter and Heather Proctor's book, and just they are two of the most enthusiastic researchers that I know of, and just seeing the world of mites, whether it's soil mites, they have a chapter on that, or whether it's arboreal mites or aquatic mites or whatever, but seeing the totality of diversity and extraordinariness through their words is a wonderful starting point.
Wonderful. So I wanted to say one more thing before we leave, which is I did look up your upcoming book yesterday, and it looks to be a special right now at Taylor and Francis. So I'm putting that link into the chat if anyone wants to peruse that. Thank you again, Valerie, for joining us today. Thank you everyone who attended, and please keep in contact with us by joining the GSBI and finding out about our next webinar. Thank you, Val.
Thank you so much, Monica and Diana. Thank you.
Bye, everyone.